There are additional impediments to the gene hunt when mental illness is the target. Diagnosis, however psychiatric manuals may try to standardize it, is often more an art than a science. Cases may be identified on the basis of ambiguous symptoms, and so a portion of the individuals in a pedigree may be misdiagnosed. These false positives wreak havoc in the mapping analysis. Another complicating factor is that the disorders are defined and diagnosed according to their symptoms, and yet it is likely that a number of genetic causes result in a similar set of symptoms. Thus, the genes underlying schizophrenia may differ from one case to the next. Even apparently clear differences between syndromes can prove messy when viewed through the microscope of genetics. Since 1957, we have known that BPD and unipolar disease, the condition characterized by depression alone, are genetically distinct syndromes. But, confusingly, there is some genetic overlap between the two. Unipolar depression is much more common among relatives of BPD patients than in the overall population. Partly for such reasons, the genetic culprits of mental illness have so far proved particularly elusive. A recent study reveals that as many as 12 chromosomes, half the total, have been shown through mapping analysis to contain genes contributing to schizophrenia. It's the same story for BPD, in which genes on 10 chromosomes have been implicated. One interesting finding is that there does seem to be some overlap between the gene regions identified in the separate mapping studies of the two maladies. Perhaps then there are some genes responsible for the overall organization and structure of our brains. Malfunctions in these genes may be the cause of the delusional or hallucinatory episodes common to both BPD and schizophrenia. The history of this research is full of high hopes brought low. A study will identify a strong correlation in one pedigree, and then subsequent research will fail to generalize the result in other populations. Such was the case in 1987, with a much publicized study of BPD in the Amish. A promising connection with chromosome 11 did not fare well in follow-up studies. Stanford geneticists Neil Risch and David Botstein have aptly articulated these disappointments. In no field has the difficulty of mapping disease genes been more frustrating than in the field of psychiatric genetics. Manic depression, bipolar illness, provides a typical case in point. Indeed, one might argue that the recent history of genetic linkage studies for this disease is rivaled only by the course of the illness itself. The euphoria of linkage findings being replaced by the dysphoria of non-replication in other populations has become a regular pattern, creating a roller coaster type existence for many psychiatric genetics practitioners, as well as their interested observers. Without denying these difficulties, I'm extremely hopeful that we are right now entering an era of genetic analysis that will soon take us beyond this irritating game of now we have it, now we don't. Two innovations hold the key. First, the candidate gene approach to finding the genes. With both the complete human genome sequence and a rudimentary functional understanding of many genes finally in hand, we can narrow our search as never before, homing in on genes with functions related to a given disorder. In the case of BPD, for example, a condition apparently connected with a fault in the mechanism by which the brain regulates its concentration of certain chemical neurotransmitters, like serotonin and dopamine, we might choose to concentrate on genes that produce neurotransmitters or the receptors. Having chosen our candidate gene, we simply compare its sequence in affected and unaffected individuals to determine whether or not a particular variant might correlate with the disorder. In 2002, Eric Lander's team at MIT's Whitehead Institute surveyed 76 BPD candidate genes. Only one proved to correlate with the disorder, a gene encoding the brain-specific nerve growth factor, the neurochemical tested as a possible treatment for Lou Gehrig's disease. But one truly relevant gene can be extremely valuable. The one found resides on chromosome 11, apparently vindicating the original Amish study, which long ago implicated the same region of the chromosome in BPD. Technological improvements underlie the other reason for my optimism about the hunt for these elusive genes. To detect the subtle effect of a particular gene, we need extra-sensitive statistical analyses, which themselves require very large data sets. Only with the advent of high-throughput sequencing and genetic typing technologies have we had the capacity to collect appropriate data for huge numbers of markers from huge numbers of people. Not surprisingly, such industrial-scale genetic analysis is beyond the reach of most academic labs. So, we will see biotech companies bankrolled by the pharmaceutical industry come to play an increasingly prominent role in this area. In 2002, two such companies, GenSet in France and Decode in Iceland, identified separate genes implicated in schizophrenia. These discoveries are a major step forward because we've now fingered actual genes, as opposed to merely mapping an effect to a region of a chromosome. We can study gene function to learn about the biochemical basis of the disorder. Strikingly, both genes are involved in regulating the function of a particular neurotransmitter, glutamate. With these new approaches, candidate genes and super-powerful genetic mapping, I am confident that we will soon uncover the major genes contributing to BPD and schizophrenia. Hopefully that will lead to improved treatments as well as to a better understanding of how genes govern the workings of our brain. For traits about whose neurochemical basis we have no clue, however, the roller coaster ride of euphoric expectations, dysphorically dashed, is likely to continue. This has often been the case in studies of non-pathological behavior. Dean Hamer's 1993 analysis of the genetics of male homosexuality provides a case in point. It caused quite a stir when he found a particular region on the X chromosome that seemed to correlate with being gay. If being gay were proven to be as much a function of genes as is, say, skin color, then perhaps anti-discrimination legislation applicable to skin color was equally applicable to gays. Hamer's finding, however, has not withstood the test of time. Nevertheless, I suspect that as we develop more statistically powerful means of analysis and learn to recognize and discount weaker correlations, we will indeed eventually identify some genetic factors that predispose us to our respective sexual orientations. But this should not be taken as purely determinist conjecture. Environment is never to be discounted, and a predisposition does not a predetermination make. My pasty complexion may predispose me to skin cancer, but absent the effects of ultraviolet input from the environment, my genes are merely a matter of potential.